is my opening slide. So I'm very excited to share from the perspective of Syntalon, allele biotechnology, and allele Cypert, some of the current advances in regenerative medicine as it pertains to healthy aging. So this is one of the major goals that we have here at Syntalon is to see if how do I advance this? Okay. So one of the I'd say one of the largest goals for our new Syntalon for Aging research is to understand how all of the technologies that we bring to bear, all the basic science research, have translational applications. And some of the biggest applications are in aging well and in health. Because of the growing population that is not only aging, we want to achieve a high quality of living for most people. And so on the right, you see a survival curve. It looks like we are living longer. People are eating healthier. People are exercising more. But there is a more precipitous decline in the quality of life for many people, especially around the world where they don't have good access to medical treatment. And some of our fondest wishes here at Syntalon is to develop something that is scalable and easy to spread across the world, whereby we can use our technology to improve health at a cost-effective manner. So those are the two biggest points: is to find the right technology and to do it and implement in a cost-effective manner. Okay, so this is a brief overview of the Syntalon Research Institute. We are 20 faculty members who come from different backgrounds. Myself, I started out in developmental neuroscience and now have interests in neuroinflammation, in iPSC generation, and in rejuvenation. How we study aging as an institute and as a group, we have a number of experts who develop different models. And in the United States, you may have heard that we're pushing towards more and more humanoid models. So we want to develop more human-based iPSC organoid cultures that allow us to test rigorous problems. We still do rely on rodent models. So whether it's mice or rats, they are still an integral part of the research endeavor. And of course, we want to translate some of those models into human disease. For us, we focus on several distinct areas. We focus on neurodegeneration. We focus on diabetes, particularly type 1 diabetes. We also focus on blood-brain barrier and how we can encourage different small molecules, drugs, uh, biologics such as antibodies or nanobodies, and exosomes to penetrate the brain to affect downstream applications. And then we also study metabolism. How does the mitochondria and energetics impact the health of different tissue organ systems? And how does that result in disease when you have diminished energetics? I will pull out a few different examples of how Syntalon has made an impact in the recent years, but it's based on our ability to create very high fidelity cells coupled with our 3D printing technology. We're also able to restore some of the hallmarks of aging, including hearing and sight, through our cell-based or our small molecule programs. And then we are also very interested in decreasing injuries to the brain from a neuro degeneration perspective. Two recent additions to our Syntalon family I have to specifically mention. is Professor Hideyuki Okano. He's Professor, Dean of Medicine at Keio University. He's the current ISSCR, or the Stem Cell Consortium Chair, but he recently joined Syntalon as a Scientific Advisory Board member. And the second person is Dr. Michael Yang. He's the ex-CEO of a company in San Diego called ViaCyte, which was purchased by Vertex Pharmaceuticals in pursuit of type 1 diabetes therapeutics. He's got a long history of innovative research and taking companies to great exits. And so now he's joining Syntalon as a board of director member. So some of the things that you may or may not have heard about Syntalon is that we have very key and important technologies. For example, one of the worldwide impacts on COVID in the past 5 or 6 years was our key technology, also with Aleo Biotech, to produce a protein, a fluorescent protein called amnion green. So if you can see over here, this is the world's brightest monomeric fluorescent protein. This is the crystal structure. And you can see that these cells, the cytoskeleton has been lit up with a conjugate for the amnion green. And why this protein was so important was because Pfizer, BioNTech, Regeneron, and others chose to use amnion green as the screening protein to accelerate their COVID vaccine and drug development. So during the pandemic, when days and weeks were precious, each of these different companies independently said they needed the best, brightest fluorescent protein to do their screening. And so all of them have chosen to use amnion green. And so that is one of our, I'd say, contributions to shortening the cost and the human lives saved in the worldwide pandemic for COVID. Another facet of our work is some of the work I've been doing for a disease called hydrocephalus. Hydrocephalus is a medical condition in young children or in adults, elderly in particular, whereby you have too much and excessive cerebral spinal fluid in the brain. And what that does is the fluid presses against the developing or the mature brain tissue, against the hard skull, and it starts to cause certain neurological problems or more brain bleeds. What's interesting is that hydrocephalus is something that many people have not heard about, but it is based on its incidence and its current rates as common as Parkinson's. So at least in the United States, about 1 million to 1.5 million people suffer and live with either the childhood forms or the adult onset forms of hydrocephalus. Over here, I'm showing you one of the molecules that might be a promising target. This molecule, this crystal structure, is a G protein coupled receptor called lysophosphatidic acid receptor number 1, and it's abbreviated LPA1. The natural ligand for this LPA1 lipid receptor, it's a fat receptor, is these different molecules down here that I've labeled 18-1-LPA, 18-2-LPA. If you look at the chemical structure, it's essentially a glycerol backbone coupled to a phosphate head group and a various length and saturated fatty acid chain. So this lipid is actually amphiphilic, so it can bind both lipid and aqueous solutions. Some of the upstream precursors of this lipid are from dietary sources. So here I'm showing soybeans, egg yolk, sunflower seeds. These phospholipids from diet are converted into a lipid called LPC, or lysophosphatidylcholine, which then gets converted into LPA, and it binds to different receptors. The reason I'm mentioning this as a important lesson is that we've understood that a lot of biology has been recognized from the lens of proteins And what's been sort of an untapped side of research has been the understanding of how lipids and fats play important roles in brain bleeds, in inflammation, and in neurological problems. So this is something that, if especially for those grad students who are looking to do additional research, is to make sure that you're not just thinking about proteins and different proteinomics, but lipidomics as a really interesting side of biology as well. Okay. One other large area that we're very proud of in Centelon is our in vitro model of the blood-brain barrier. So one of the professors, my colleague, Dr. Irene Peterson, is looking at large-scale blood-brain barrier models that allow her to study the endothelial cells, the tight junctions, as well as the brain side, meaning the neurovascular units, the ensheathing endothelial cells, pericytes, as well as the microglia that are typically adjacent. And in studying this, we're very interested in controlling the passage of small molecules all the way through cellular components, such as exosomes and mitochondria, through the BBB in order to affect therapeutics in the brain. So this is another area I'm happy to talk more about. This is a slide that Dr. Young showed earlier, which is what is the basis for some of the advantages that we have from alleles perspective for using messenger RNA for reprogramming. And here we've outlined several different key important ideas, which is that using messenger RNA reprogramming instead of DNA-based or viral-based reprogramming, you have very high efficiency and you have no nuclear events, which means that you don't get spurious integration of different DNA-based elements into the genome. So you don't get random insertions and things like that. The other important thing, of course, is that the messenger RNA, the signal itself is transitory. So there's a limited lifespan, and it makes it quite ideal for bringing a very digital cell fate change to the different stages. So for example, if you're going from a fibroblast to an iPSC, and then from an iPSC to a defined cell fate, the type of effects that we typically see are very digital, stepwise change in markers, typically on the order of 90 to 95% or greater. So that confers a huge advantage over other DNA, viral, or uh, growth factor-based reprogramming. Okay. One of the things that we did start to work on, and many people have already done some work on, is the idea of using messenger RNA to get partial reprogramming. And partial reprogramming essentially is taking a cell, for example, fibroblasts, and not taking it all the way to a defined cell fate, such as iPSCs, but rather using some of the Yamanaka factors, OSKM or OSK, to take the cells partially. And that partial rejuvenation uh, essentially wipes away some or many or all of the epigenetic marks that are on the DNA. And by wiping away some of those epigenetic marks, you essentially force the cell to be a younger cell. Now, in alleles case, instead of the original Yamanaka factors, the OSKM, or these, you see OSK, OSK plus, we use 6 factors. And we use, obviously, the messenger RNA system. So I just want to show a couple of data slides. It won't be too data heavy. But what one of my lab members was able to do was to compare 3 different cell lines from 3 different individuals. So line 1 was from an individual who had 1 or 2 millimeter skin biopsies, who was an older gentleman, average fitness. And we won't go into details what average fitness is. But the comparator is aligned to somebody who is older but high fitness, somebody who was regularly running marathons. And then line 3 was somebody who was in their teens, younger and high fitness, so somebody who was in sports. And so we interrogated some of their baseline cell cycle characteristics of these cells, and you can see the G1 phase peaks, the S phase, and the G2M. So just so that you can see that there is already high heterogeneity amongst different people of different fitness levels and different identities. Now, we didn't do an exhaustive study to look at males and females and all the different age groups, but my suspicion, if we were to do this at high scale, would be that you would see very distinct cell cycle curves for many people. What we then did was we took the different cell lines, these fibroblasts, and we treated with the 6F mRNA cocktails that we had. And we, down here, you can see that we treated the cells once daily for 3 consecutive days. And you can see that at least here, we're seeing quite significant changes in the S phase. So there was an increase of cells that displayed S phase, not in the G2M and not very much in the G1 phase as well. So the idea is that this process of using and exposing these fibroblasts, these different lines to messenger RNA, allowed you to increase DNA synthesis. Okay. Okay. Now, that was something that we had done a couple of years ago, and then we have a new colleague at Syntalon, Dr. Alexei Turskik, who then published a paper in Nature Aging about a year and a half, 2 years ago. And he uses a high throughput imaging technique to quantify aging and rejuvenation. So what they did was they isolated nuclei. These days, a lot of people use isolated nuclei for downstream omics, RNA-seq. What they did was they took nuclei from these different organs of mice. Then they stained for different markers, epigenetic markers. So for example, this one was histone H3, K4 methylated 1, histone H3, K4 ME3, or histone H3, K27 acetylation. So using these different markers, they were able to segment the nuclei based on the staining. And I won't go into all the details of how they mapped out the nuclear territories being defined by these 3 marks, but they were able to extract a significant amount of distinguishable features between young and old animals. So this is one example of the data that you might see or that they saw was they were able to induce using doxycycline the 4 different Yamanaka factors. They took liver and muscle, and then they used their image analysis or nuclear segmentation using these histone immunofluorescence markers to segment. And they were able to show quite convincingly that the young mice had very distinct and different profiles compared to the aged mice that were given the OSKM induction versus the control, which is the aged mice. So right now we're in the process of repeating some of the experiments we've done using the allele factors, but using this strategy for assessing rejuvenation. So how far are we taking the cells and rejuvenating using the partial reprogramming strategies. We're also putting in different inputs. So we can use either mitochondria, so frozen or fresh isolated mitochondria or exosomes to infuse into old cohorts of mice. And we hope to look at some of these readouts over here on the right. This is an area I'd love to talk to many of you about in terms of how we can collaborate. But we believe that we can look at least grip strength, their mobility in open field, their coordination using rotorod, memory using some of the maze tests. And then we can interrogate using bloodborne factors to look at SASP, other factors, look at their circadian status, looking at their nuclear epigenetics, and as well as tissue architecture. So this is by no means an exhaustive list. There's many different readouts depending on how you perceive aging to be. But the important thing is that we have this kind of pipeline that we believe it allows us to test in a rigorous fashion some of the upstream, what we think are perturbants, things that can perturb aging and rejuvenation, and look at the rodent readouts. Of course, we'd love to do this in humans at some point. Okay, so this just is a quick slide on the number of different cell types that many others have reported being able to grow from iPSCs. And there's a number of different diseases that are very relevant using cell-based therapies. And so I'm going to focus on type 1 diabetes in particular. Before I go into that, this is just a very interesting slide I saw at the ISSCR in Hong Kong in the past year. This is work from Kyoto University. The idea is that we can treat Parkinson's disease by implanting, injecting, or transplanting the right dopaminergic cells into the substantia nigra of patients. And so a number of clinical studies are underway in Japan and the United States. Here, in fact, Aspen is in San Diego, and others to try to treat Parkinson's. I believe we're now in phase 2 clinical trials for this strategy. Okay. This is, of course, something that Dr. Young also alluded to, which is that we are able to start with a small 1 or 2 millimeter biopsy to essentially grow unlimited amounts of iPSCs or propagate them to additional fibroblasts using our messenger RNA technology. And there's numerous potential therapeutic applications. I think if you were to read or search on either PubMed or other databases, you can see that many clinical trials, including clinicaltrial.gov, there's many people using fibroblasts or MSCs or some variant of those cell types to go after macular degeneration, heart repair, arthritis, and other areas. And some of this is dependent on the ability of some of these cells to modulate inflammation or reduce inflammation in these areas. And others, it's to encourage the existing cell types in those tissues to expand. This is just a quick screenshot of the number of companies that are involved in iPSC-derived cell therapies. Of course, this slide is a year old, so there's more companies, and they've adjusted based on funding and their clinical trial statuses. But this is very encouraging because as of 10 years ago, this list was much shorter. So from 10 years now, there's a exponential growth in these number of companies, and I believe it's because of the breakthrough from Yamanaka and others, as well as maybe some contribution from ourselves to open the gates wide for iPSC-based cell therapies. The ones here I've circled in red are some of the ones that Allele has specifically worked with, Sonetta Therapeutics, Heartseed in Japan, SCM in Korea, of course, the National Institutes of Health here in the United States. This is a worldwide map, and we hope, really one of our fondest hopes and wishes is that we increase and tighten the collaboration between Aleo Biotech and numerous medical institutions, companies in Taiwan. So we believe that this will be a very fruitful area going forward for many years to come. I wanted to show you a couple of slides. So with alleles reprogramming technology, at day 3, this is what you see after we do a 1 to 2 millimeter skin biopsy. And over here, these are some of the patents that Allele has been awarded. The strengths of our technology is that it's quite fast. So our strategy takes 6 to 9 days to get to iPSCs. It's very robust and reproducible, meaning that we've tried to grow more than 200 passages and 200 runs. So we've done distinct patients, and we've done more than 200 distinct creations of iPSCs. And we've taken those iPSCs and passaged them more than 250 times, and they remain stable. So some of you who work with iPSCs and stem cells knows that in the past, there are many different techniques for trying to prevent iPSCs from differentiating. And so what we believe at Allele is that the mRNA reprogramming gives you high quality cells to start with, so you don't have differentiation problems. Typically, it's through culturing or other processes. The GMP process is quite robust. We've made numerous cell lines, and it's xeno-free and no viruses, no genomic integration, as I mentioned earlier. Over here, this is day 8. So from day 3 to day 8, you can see that there's significant growth and morphology, and that now begins to resemble your iPSCs. For Allele, these are all the specific cell types that we've been able to make. And in fact, the list is going to grow longer because I'm going to now mention a liver project that we just created 14 new cell types. So this is the old list, and we have a new list now. Okay, so I'm only going to go into 2 distinct diseases. So first is that we have an internal program where we try to treat type 1 diabetes using iPSC-derived beta cells. We internally call this our AI beta spheroid program. And Allele has been able to produce and scale the production of beta cells from iPSCs to the tune of 2 billion cells at each given, I would say, at each given production. So we can keep doing 2 billion, 2 billion, 2 billions. We've also done some in vitro testing. So we've done glucose stimulation, and we've shown that the in vitro stimulation of these cells induce them to produce human insulin that's robust and functional. And then these spheroids are quite similar in size to human islets. And so you can see that we've got the scale bar at 200 microns. So we believe that the spheroids are about 50 to 150 microns in size and consistency. So this is important because for our subsequent animal models, we do implant these to show that they are incapable of reducing mouse diabetes. On the right, it's not going to be too data heavy. I'm just showing you a little bit of flow cytometry data of some of the markers that we look for to convince ourselves that our differentiation protocols are very robust. For example, our PDX1 marker is at 85. Insulin is at 98.5%. C-peptide is about 99.9%. NKX, transcription factor, is 95%. So we do believe that we have a very digital way of taking iPSCs and converting them into end-use beta cells. This is just a sample data of some of the mouse cohorts that we've run. So we have induced type 1 diabetes in mice two ways. One's we use a drug called STZ, or streptozotocin. This drug selectively kills the beta cells, and it leaves the mouse. So here, when you start administering this STZ, particularly into male mice, you get a very reproducible increase in glucose levels. So the mice are up to 500 mg per deciliter of glucose, of blood glucose. And as we start to implant our beta cells, we can start to see a decrease and drop in the glucose levels. At the same time, we are also measuring the insulin production. We've had these mice now for about, this data is old. We've had these mice out to be about a year and a half now, and we still detect human glucose being secreted. So we believe that when we sacrifice these animals, that we're going to see quite robust engraftment of these beta cells. We implanted them through the portal vein of the liver. So we believe that when we do our histology, we shall see quite a bit of those iPSC-derived human beta cells. On the right over here, this is a graph showing the blood glucose levels after we gavage. And so in a typical challenge, you'll see an increase in peak and then should come down as the mouse starts to produce its insulin. So in a normal mouse, the mouse insulin will cause this type of curve. In our STZ-induced diabetic mice, it's the human insulin that is produced by the human beta cells that's causing this curve. Okay, so now this is the final project that I want to tell everybody about. And again, this is the Moonshot project that Dr. Young alluded to earlier. I only have a few more slides, so thank you for your patience. So this one, it's called a moonshot because like DARPA, US's military, advanced military agency, ARPA-H is aimed at doing moonshot projects for health. And our current funding is through a mechanism called the PRINT program. So the PRINT program is something where the goal is to produce an entire transplantable liver, 240 billion cells in a reasonable amount of time. In our case, the grant is going to run for about 5 years. So one of the successes so far to date is that we've been able to produce 14 different cell types of the liver within the first 3 months of this grant. So prior to this grant, we had no knowledge of how to make hepatocytes or liver sinusoidal endothelial cells or cholangiocytes. But through some of the reading and research that we have done, coupled with the speed of the messenger RNA, we've been able to start making these cells at scale and at quantity. So that's to say that we've actually performed ahead of schedule. And right now, what we're doing with our collaborators at UC San Diego with Xiaochen Chen is we're using artificial intelligence and AI-driven analysis to look at the microstructure of liver so that we are able to understand how to 3D print these different 14 cell types into the correct configuration, whether it's a liver mini organoid or other microstructures. So this is an example of some of the printers that we use. We did incubate a company called Allegro 3D that was later purchased by CellLink, and they created the BioNova X, which is to print cells and some sort of matrix, in our case, an alginate matrix to produce structures. So here we were being a little playful. We printed the allele logo using the alginate. But in the future, we'll be able to encapsulate cells, beta cells, or other cells into this alginate material and produce the right microvasculature, create the right structure. So I know I've talked a lot and there's a lot of detail, but I wanted to just summarize 5 points for you. And I think this is a very hopeful summary slide. I believe that messenger RNA has become mature enough, whether it's through the pandemic COVID experiment or all the subsequent work that has been put into it. But messenger RNA has become a very robust and reliable technology to take and reprogram your initial cell types, such as fibroblasts, into induced pluripotent stem cells and then create the needed cell types. And we can do this at cGMP levels and factory scale. The second is to use messenger RNA and to interrogate how far we can program and reprogram cells to alter their epigenetic state, whether it's to completely wipe out their full epigenetic state or to partially change their epigenetic state. The third is that we now can take and test input perturbations, put messenger RNA or mitochondria or exosomes or other cells and look at different readouts in intact organisms to assess whether we really are changing aging at a fundamental level. Number 4, I think, is part of number 1, but really that we can create high purity cells so that you don't have this Gaussian distribution of cell fates that creates a lot of cost. And then finally, we at Allele have taken the mRNA technologies and we've worked with TriLink, and we believe we've gone beyond some of the clean capping technologies to allow very efficient rapid generation of different cell types. And it allows us to really shorten our protocols, decrease costs, and increase the robustness of our strategies. So I think that all of that is being tested and borne out through the Liver project. And finally, I just wanted to end with a slide. Obviously, it takes a whole group of collaborators and friends to do all of this work. So some of the work that I've been doing is through my lab and collaborators, faculty at Syntalon, of course, through Allele Cypern and Allele as well. Special mention to Dr. Young and Michelle Yang. And then for the hydrocephalus work that I wasn't really able to mention, we're being supported by different awards and a huge list of collaborators who are very intent on trying to solve this current surgical problem, make it more of a drug-based solution. And then finally, we did do some work with UC San Diego and the Deerfield Fund to create dopaminergic cells for treating Parkinson's. And finally, our latest project is this ARPA-H print project. It's a very large project and to the tune of $25 million to get this done in 5 years. So I really appreciate everybody's attention. Thank you so much.